I'm going to do something slightly unusual at the beginning of my talk, which is to show a 2-minute promo video for the book. It serves two purposes. One is to sort of give you a hint about what the book is about, to give us a starting point. But the other, and perhaps more important thing, is I want to showcase the wonderful work of Anne Odling-Smee and her team at Design Science, who produced this promotional video, but not just that, also produced the Evolution Evolving website and all of the social media platforms that we use and many of the posts, as well as the wonderful animations that you can find on the Evolution Evolving website. If you haven't had a chance to look at the website, please do, because these, I think these animations, they're usually about 5 minutes. They're a wonderful educational resource in and of themselves, and you know, a terrific little introduction, a very attractive little introduction to scientific concepts like plasticity-led evolution or the role of developmental bias in evolution. So, without further ado then, This is an exciting time for evolutionary biology. New scientific discoveries are pouring out of the research laboratories, challenging traditional understanding. It turns out that whales have cultural traditions that are driving genetic evolution, that rats adapt to poisonous diets through exploiting the genes of bacteria they get by eating feces and soil, and that learned fears can be inherited across generations through molecules that attach to DNA. Phenomena that scientists used to think are impossible, such as the inheritance of acquired characteristics, have recently been shown to happen. Newly discovered mechanisms like epigenetic inheritance are making scientists think again. Now, I've got together with a prize-winning group of leading biologists from top universities around the world to write a book that communicates some of these new discoveries. Written in a non-technical style, Evolution Evolving captures the emerging idea that how organisms develop, including their behavior and their plasticity, and what organisms do, such as the changes they bring about in their environments, influence the rate, pattern, and direction of evolution. Our book shows that there's far more to biological inheritance than genes, that living organisms are in fact communities, and that populations evolve along corridors created by developmental mechanisms. This has major implications for understanding human evolution too. Humans didn't evolve like mice or fruit flies. We interpret natural selection differently because we develop differently, we interact with the world differently, and we inherit selectable variation differently. Read Evolution Evolving today and be inspired with a new vision of adaptive evolution. I hope you're all feeling very inspired. Okay, so our understanding of evolution, particularly adaptive evolution, is of course dominated by natural selection. Perhaps one thing that is distinctive about Evolution Evolving is the suggestion that natural selection is not just something that happens to organisms. Their behavior, their development, their activities influence whether and how it happens. That means the form that natural selection takes depends on the individual properties of organisms. And hence, as organisms evolve, then the evolutionary process itself evolves. So our book is organized into 4 parts. There's an introduction in which we make the case that one needs to consider development to understand evolution. We then provide a brief précis on how development works. We go on to describe how developmental processes affect evolution, and then consider in the final section some implications of that. The opening chapter is entitled "Nothing in Biology Makes Sense Anymore," which is obviously a kind of play on de Bianchi's famous quote, "Nothing in biology makes sense except in the light of evolution," and makes reference to the fact, as we describe in that chapter, that evolutionary biology in recent years has been subject to a rush of often striking new findings that, for many of us at least, seem to challenge the orthodox narrative about how evolution works. So much of that chapter is taken up with describing some of those examples which are making people think. In later chapters, we go on to describe something of the history of the field of evolutionary biology and try to account for why development might have been left out of the evolutionary synthesis that was forged in the last century, as well as describe some early developmental perspectives, such as the organicist, best known of whom is Conrad Waddington, who is one individual to whom we dedicate our book. And we also go on to talk about some major debates which are taking place within the field of evolutionary biology and how developmental processes might be central to those. But what's really fun about our book, I like to think, are the empirical examples. So let's talk you through a few examples. So we begin with the fascinating case of the Mojave Desert wood rats that feed on these highly toxic creosote bushes. And they consume vast quantities of creosote, a toxin, every day. And effectively have opened up a new niche for themselves, largely free of competition, because they were able to process this deadly food. What's really fascinating about this case is that their capacity to consume this toxic food is entirely dependent on the bacteria inside their guts. When researchers gave these wood rats antibiotics that wiped out their bacterial community, they rapidly declined on this diet and would have died. Conversely, when they took wood rats from other regions that normally don't feed on creosote and would normally be poisoned by it, and inoculated them with a bacteria taken from the Mojave Desert wood rats, they were able to thrive on the creosote diet. So here the microbiome, which is of course the collective noun for the array of bacteria and archaea and fungi and protists and all those tiny little organisms that reside inside and on the surfaces of larger organisms, pass down the generations externally in the external environment and are picked up each generation anew with the wood rats consuming soil and feces. So these Mojave Desert wood rats have exploited this novel foraging niche, it's thought for hundreds of years, through the stable ecological inheritance of their externally acquired bacteria. Now let's move to the other side of America, and to the East Coast, where in the Gulf of Maine, a population of humpback whales has also opened up a novel foraging niche for itself. These whales feed on sand lance, which are an eel-shaped fish that form these huge shoals, through a novel foraging method known as lobtail feeding. This entails an individual whale slapping the water surface with its tail fluke, which shocks the fish below into tightening their school. It then swims around the school of fish, releasing air from its blowhole, which traps the fish in a net of bubbles. It's called a bubble net. And then they swim underneath and up with their mouths gaping to feast on all those trapped fish. This is a highly productive behaviour that was first observed in 1980, and researchers can identify individual whales. They were able to establish that this behaviour spread from a single individual to many hundreds of whales in the region, and that this behaviour is learned and culturally transmitted. So this behaviour has been passed down the generations, independent of cultural transmission, for many decades through cultural transmission. And we can also see evidence in other cases that this culture is affecting genetic evolution. So for instance, in killer whale populations, otherwise known as orcas, there are clans or groups that feed on different foods. Some feed on fishes, others feed on seals and sea lions, others feed on dolphins. And once again, it's well established that these are learned and culturally transmitted foraging behaviours. But also they have modified the natural selection acting back on those populations, selecting for morphologies, head shape, teeth shape, digestive physiologies that are well suited to their learned feeding habits. So in this case, culture seems to be imposing a direction on genetic evolution. Well, we describe many such examples in evolution evolving. And They excite controversy, of course, because of their implications for our understanding of natural selection, which Richard Lewontin, who is a second of three individuals to whom we dedicate our book, famously decomposed into three component parts or requirements. One is that there must be trait variation among individuals in a population. A second is that individuals with some variance must leave more descendants than others. And a third is that offspring must resemble their parents more than they resemble unrelated individuals. Now you'll notice, of course, that these are all fairly abstract statements. These requirements are met, then natural selection will ensue. But of course, evolutionary biologists will have a particular way of thinking, a particular implementation of these requirements, which is generally thought about in genetic terms. So we'll tend to focus in on inheritance as the transmission of genes. We'll think about phenotypic variation, or at least evolutionary significant phenotypic variation, as ultimately reliant on mutations in genes that occur at random with respect to function. And of course, natural selection acts on genetic variation. There are other assumptions that are commonly made, but perhaps less explicit. One is that these sub-processes are assumed to be largely independent, what Walsh calls quasi-independent. That is, they feed into each other, but they don't affect each other's operation. So, for instance, we might assume that big beetles will outcompete small beetles, and that will influence which beetle genes are passed on to the next generation. But it's not generally thought at least, that will change the very nature of Mendelian inheritance. So when we take into account that inheritance is generally unbiased and that, as I say, phenotypic variation results ultimately from random genetic variation, that means that all of the direction in adaptive evolution comes from differential fitness for a particular population, a particular environmental context. And as a consequence, we can think about all organisms evolving in broadly the same way. Now, the new data that we present in Evolution Evolving actually challenges all of those assumptions. Think back to the whales, for instance, where cultural transmission is not just a source of phenotypic variation, not just a proximate mechanism, but it's also changing the very nature of inheritance. Inheritance goes from just genes to genes and culture. And that cultural transmission, as we saw in the killer whales, appears to be imposing direction on adaptive evolution. Now, of course, these issues would not be contentious if there weren't ways of sort of rescuing a traditional explanation within evolution. For instance, by characterizing such examples as I've described as rare or idiosyncratic or short-lived, temporary in their influence or under genetic control. All of those arguments have been made and more. We suggest instead in Evolution Evolving, we should embrace those examples and a developmental perspective. We suggest that there's a framing or a way of thinking about evolution by natural selection that makes developmental processes causally important for evolutionary adaptation and diversification. A framing that not only provides better explanations, but can inspire different kinds of experiments and theory and open up new research questions. In the second section of our book, we focus in on how development works. And our goal here is not to provide a comprehensive account of the development of multicellular organisms. Scott Gilbert has already produced a market-leading developmental biology textbook. Rather, what we want to do is to give you a kind of little taster of really what's important about development that you need to know in order to do a good job in understanding evolution. This leads us to 5 general principles that we emphasize are important in evolution. That development is modular, that it's epigenetic, by which we mean it occurs through interactions that occur above the level of the gene. That it's constructive, so we can think of the organism as an active agent that responds to and modifies both internal and external states. That's interchangeable with respect to genetic and environmental inputs. Whether there's a mutation or a change in the environment, they'll frequently be processed by the same developmental machinery and commonly lead to similar phenotypic outputs. And that it's prone to generate biased phenotypic variation. Biased variation that can itself influence the direction of natural selection. Now, I've talked a lot about development. I want to emphasize that when we use the term development, we're talking about the entire life cycle, not just what happens early in that life cycle. So it includes things like plasticity and learning and culture and niche construction. It also includes developmental mechanisms such as exploratory mechanisms. Now, like Mary Jane West Eberhardt, who's the third of the three people to whom we dedicate our book, exploratory mechanisms are something on which we place particular emphasis. They're a form of developmental mechanism that operates on kind of Darwinian principles through generating adaptive phenotypes iteratively, producing variation often at random, testing what works, testing performance, and then regenerating or retaining the things that work whilst losing the things that are less valuable. You'll all, I expect, be familiar with adaptive immune systems which operate on those kind of principles, but actually similar principles that operate in the development of the brain, of learning, the formation and growth of blood vessels, of plant roots and branches, of the cytoskeleton, and exploratory behavior of many animals, including ants. What's quite interesting about these mechanisms is they confer on organisms a degree of adaptability because they're able to produce functional phenotypes even in the face of mutation or injury or sometimes quite dramatic changes in the environment. And in later chapters of Evolution Evolving, we show how exploratory mechanisms are able to sort of account for much of the adaptive design that we see in adaptations that can then be subject to natural selection and provide organisms with a shortcut to adaptation. Okay, so part 3, I suppose, is really the heart of the book, where we focus in on the developmental bases of evolutionary processes. And we place particular attention on developmental bias and how it can result in natural selection being channeled along pathways or highly selectable dimensions of phenotypic variation comprising clusters of co-evolving traits. What links these traits sometimes is developmental regulation. They're all connected by being reliant on the same developmental mechanisms. But we show also that plasticity and niche construction can lead to connections or co-evolution of different traits and generate developmental bias. So developmental bias is the idea that certain phenotypes are produced more readily than others in development, and that bias in the distribution of phenotypic variation can affect the direction of evolution. We discuss in Chapter 7 the example of domestication syndrome. So it's no less an authority than Charles Darwin, who first pointed out that mammals, domesticated mammals, share a rather curious set of traits. So that horses and cows and sheep and rabbits and dogs and pigs should all be tame, of course, made sense. They've been artificially selected to have that characteristic. But why should so many of them have splotchy skin colour? Why should they have floppy ears or flattened muzzles or small teeth or small brains? Darwin was unable to explain that conundrum, and neither, for instance, was any scientist for the next 100 years or so to any degree of satisfaction. What did those things have in common? Well, it was actually only in 2014 that, to my mind, a satisfactory explanation came to light. What these things have in common is an origin as neural crest cells. Now, for the non-biologists in the audience, neural crest cells are embryonic cells that migrate around the body. They literally move and then differentiate into a wide variety of different cell types. And what's relevant here is that they're either direct precursors of or play critical roles in the development of virtually all of the traits associated with this domestication syndrome. So what all these traits have in common is they're reliant on the same developmental mechanism, which means it's hard for anyone to evolve independently without eliciting evolutionary change in others. A mutation in one is likely to affect others, and so we see this co-evolutionary pattern. And this is likely to, this has been shown to affect humans too. So humans have thin gracile bones, they have small teeth, they have reduced sexual dimorphism, neotenous features such as flat faces with a forehead and no muzzle, all of which can be explained through developmental bias. We don't need to have separate evolutionary explanations for each of these characters. We can understand them as evolving together. But what's kind of interesting is that there's a bias to the patterns of variation that are generated, such that it's easier to get certain combinations of these traits than others. For instance, it would be hard to evolve or to at least to achieve an animal that's tame with large ferocious teeth. We go on in chapter 8 to describe how developmental plasticity can generate developmental bias. And when it does so, sometimes we see what's known as plasticity-led evolution, where plasticity comes first, phenotypic change comes first, and genetic change follows in its wake. A really nice example concerns these blind Mexican cavefish. So when these cavefish were first discovered in the 1930s, they looked so strikingly different from any known fish that they were classified as an entirely new species, only for researchers subsequently discovered that sighted versions of the very same species were actually widespread in all the rivers and streams in Mexico and many parts of the USA too. There are now over 30 populations known to science, mostly eyeless, with reduced pigmentation, but also, again, with a suite of characteristic traits. Changes in bone structure, changes in foraging behavior, and some odd features too, such as fragmentation of some of the bones, which doesn't make obvious adaptive sense. What's really interesting here is that researchers in recent years have taken their cousins, the sighted riverfish, and raised them in the dark and in the cold to simulate the cave environment, and found that when they do so, these fish develop to have phenotypes that are strikingly similar to the cave fish phenotype. And that the traits that respond plastically to that dark immersion exhibit greater phenotypic variation upon which natural selection can act. So in other words, what these derived cavefish traits have in common, what links them, is that they all respond, or most of them at least, respond plastically to the dark. We can see a similar picture with respect to niche construction, which in this case links together traits associated with environmental modification with plastic traits that are sensitive to that environmental modification. And hence they evolve together. In chapter 8 of our book, we investigate the interesting case of dung beetles. And I'll tell you the story that we describe in that chapter. So the mother dung beetles will manufacture a brood ball out of dung in which she will insert a single egg together with what's called a pedestal, which is a little sort of package, a birthday present for the developing young, full of her symbionts. And then she'll bury that safely underground. That's her niche construction done. And then the offspring will engage in its own niche construction. It will, when it hatches, eat the pedestal and in that way acquire the maternal symbionts. It will then manufacture its own pupation chamber. It will process the brood ball in various different ways, which redistributes the symbionts throughout the brood ball in ways that allow them to process the dung themselves and make carbon nutrients available to the developing young. Now, really clever experiments done by researchers have either permitted or cancelled the effects of both the maternal and the offspring niche construction to see what difference it makes. It makes a big difference to the development of those larvae. They wouldn't have anything like normal development without that niche construction. But moreover, that niche construction is clustering those traits so that they co-evolve together. It's influencing which traits become strongly associated with biological fitness and thereby influencing the direction of natural selection. Now let's go back to this algorithm that we began with, Lewontin's 3 requirements for natural selection, and see how it's actually operating in the case of the beetles. Well, the first thing we notice is that inheritance is not solely down to genetic inheritance, because there's this really critical aspect of ecological inheritance, the inherited, modified and manufactured brood ball with its associated symbionts that it gets through the pedestal, which is absolutely critical to normal development. And secondly, we can see that the pattern of phenotypic variation that's generated is also critically dependent on that ecological inheritance. Not only is different phenotypic variation manufactured through that niche construction, but the relationships between the different traits, the extent to which they're correlated, hangs critically on that ecological inheritance. And we can also see, thirdly, that the contribution of these traits to fitness depends on the properties of the brood ball. And that means that the direction in which the population evolves is contingent on that niche construction. What are the ramifications? Well, once again, we can see the sub-processes are not just feeding into each other, but they're affecting each other's operation. But more to the point, the form that natural selection takes depends critically on the properties of the organism. This is a very beetle-specific interpretation of natural selection. It wouldn't work for yeast or killer whales. If it was killer whales, we'd have to strip out the ecological inheritance and add in the cultural inheritance. Each organism has its own interpretation of natural selection. Or as we write in our book, the activities of living creatures shape the relationship between themselves and their environments and thereby determine which phenotypes are fit and which are not. Selection depends on what organisms do. In chapter 10 of our book, we turn to extragenetic inheritance. Now, this is a topic in which there's been an explosion of scientific interest in recent years. Vast amounts of data generated on topics like epigenetic inheritance and cultural inheritance. Each of these pictures represents a particular form of non-genetic or extragenetic inheritance that is now known to science, operating alongside genes. We try and make sense of this vast amount of data in our book. And we can organize this data into 4 pathways, if you like. One is germline transmission, which applies to not just genes, but also structural inheritance in single-cell organisms where the actual physical properties of the cell act as a template, which then allows other cells to be copied and then produce similar characteristics. There's epigenetic inheritance, for instance, resulting from DNA methylation patterns and other chromatin marks that are passed across the generations. Second pathway is from the parent's body directly to the germline, which occurs, for instance, in the case of the uptake of proteins from the parent's blood into the eggs in many birds, but also the transfer of parental RNAs into the germline in nematode worms. And then there's a whole rash of things that are being passed from parents to offspring, from body to body, things like the transfer of symbionts, bacteria, archaea, protists, fungi, viruses, transfer of hormones, nutrients, antibodies, prions, other forms of epigenetic inheritance, cultural inheritance. And this, of course, is another domain where there's vast data. Hundreds and hundreds of species of animals have been shown to be capable of cultural inheritance now, not just vertebrates, but invertebrates too. And the fourth category of ecological inheritance, or body-to-body transmission indirectly via the external environment. And we saw an example of that with the first example I gave you of the wood rats that are acquiring their symbionts indirectly by eating feces and soil in their environment. Now, for over a century, this idea of soft inheritance, which is the idea that heredity can be affected by the individual's lifetime experience, was regarded as highly disreputable. Ernst Mayr famously said it was a chemical impossibility. Now, in the 21st century, we see that it's everywhere. And the challenge really is that we're swamped by data. What do we do with it all? Why are there so many different forms of inheritance? Do they work together or separately? What job do they do in evolution? Well, in chapter 10 of our book, we provide answers to those questions, but we point out it requires us to think slightly differently about adaptive evolution and suggest that many extragenetic inheritance processes are best regarded not as they've been traditionally viewed, as noise or fine-tuning or baroque add-ons, but as essential tools for short-term, rapid response adaptation. Fourth section of our book is concerned with some of the implications of this developmental perspective. Implications for the origins of evolutionary novelty, for the evolvability of organisms, for the understanding of the human condition, and for understanding the nature of evolutionary theory itself. And just to give you one illustration of the kind of implications we're talking about, let's consider the evolutionary implications of the existence of epigenetic inheritance. Now again, for the non-biologists in the audience, I'll explain how the DNA in your cells, indeed in the cells of many multicellular organisms, is not typically naked, but it's often clothed with chemical attachments which will influence whether nearby genes are expressed and the extent of that expression. And when those cells are copied and new cells are made, often that machinery is also copied, and that's how you can get cell lines. And sometimes it persists through the production of the sex cells, sperm and the eggs, and into the embryo. So you get transgenerational epigenetic inheritance. Now, this is not a rare phenomenon anymore. There are literally hundreds and hundreds of examples, perhaps thousands of examples, documented examples of epigenetic inheritance in a very wide array of organisms. You can see some of the organisms illustrated in the bottom row there. Now, this figure comes from a review of 500 published papers, each reporting examples of epigenetic inheritance in a wide array of organisms. And what it shows is how long that epigenetic inheritance is persisting across generations. Now, you can see there's one row with the blue line, which represents nematode worms, where epigenetic inheritance has been shown experimentally to persist for over 20 generations. But in the vast majority of organisms, these effects peter out in 3 to 5 generations. But that does not mean they're not evolutionarily important, as we show in our book. To give you a really simple and straightforward example of that, this is a lovely experiment carried out by researchers on Arabidopsis, which is a small flowering plant which is widely used in genetic studies. You can think of it as the sort of lab rat of the plant world. And it's been established that many of the traits of this plant, flower shape, flowering time, root growth, root density, are influenced by patterns of epigenetic variation. And these researchers carried out an experiment where they selected on a variety of different phenotypic traits associated with Arabidopsis, and after 5 generations found good responses to selection, so the traits changed over time. But that was linked to changes in epigenetic variation rather than genetic variation. The initial response was on epigenetic variation. And they conclude, thus we propose that epigenetic variation is subject to selection and can contribute to rapid adaptive responses. So data like these, or I should perhaps say other studies, have gone on to look in other species at the effects of cancelling or not having that epigenetic variation. Very frequently, such populations without that capacity to evolve very quickly in the short term go extinct. So these type of data challenge us to face up to encourage us to face up to some real challenges. Challenges in how we think about and understand natural selection, where it would seem that genes are not the only form of heritable variation subject to natural selection. There's now substantial evidence for adaptation occurring through the selection of epigenetic, symbiotic, and cultural variation. Challenges in how we understand evolvability, the ability of a species to evolve. If evolvability is defined as it commonly is, the ability of a population to respond to selection, an adaptation that occurs through the selection of extragenetic variation needs to be considered. But thus far, that's been given relatively little attention. And even how we think about evolution. For a century, evolutionary biology has been preoccupied with the slowly changing aspects of evolutionary adaptation and the most stable components of inheritance, i.e. genes. Now, in the 21st century, it's time to recognise extragenetic inheritance as playing a vital and central role in rapid evolutionary adaptation. So the selection of epigenetic, symbiotic, and cultural variation in particular is not analogous to biological evolution. It is biological evolution. It's the fast part of adaptive evolution. And this has implications for how we think about human evolution too, because humans, of course, have got an intensely powerful form of extragenetic inheritance in our culture, a form of extragenetic inheritance that has powerful influences on many human phenotypes, that influences human fitness in a variety of different ways, and which underpins our extraordinary capacity for niche construction to modify our environmental states. Humans have been characterized as champion niche constructors. And for that reason, it's because of those kind of characteristics I was able to claim in the voiceover to that promo video with which we began that humans evolve in a different way from fruit flies and yeast, because we develop differently, we interact with the world differently, and we inherit resources differently. So, human evolution is an undeniable scientific fact, but it involves more diverse and complex processes than are represented in most treatments of Darwinian biology. Moving on then finally to the last chapter, we consider some of the implications for evolutionary biology itself. And here we reflect on the fact that some researchers have characterized the work of the field of evolutionary biology as largely done. But when it comes to our developmental perspective, we see bright opportunities for important new research. For instance, once it's recognized that developmental biases can lead to biased genetic and phenotypic variation, then understanding the characteristics of that variation, trying to explore to what extent the investigations of developmental mechanisms would shed light on the nature of that bias or make sense of patterns of phenotypic variation that we see out there in the world becomes central questions for evolutionary biology, as do trying to understand the developmental origins of fitness differences or how different inheritance streams interact. If evolutionary biology is to discover the mechanisms by which new phenotypes come into existence, succeed and diversify, mechanisms in which developmental processes play key roles, then we conclude the field is only just getting started. So I'd like to thank many people who've helped in the production of this book, and that includes several people that I can see in the audience, so thank you all for your feedback on chapters and helping in a variety of different ways. To mention, if you want to get hold of the book, please do pick up one of these bookmarks, which will give you a 30% off, or just take a bookmark if you need a bookmark. We've got loads. And I'm going to end by quoting what is the very last sentence of the book. Once it's accepted that the evolutionary process evolves, it follows evidently that developmental mechanisms must matter to evolution. And conversely, it's because development matters that evolution is evolving. Thank you.